Welcome, everybody. Thank you for joining us. This is our annual event of the ALS Therapy Acceleration Program. The Therapy Acceleration Program of ALS, in short, TAP, is a pioneer of venture philanthropy. Today, the focus is on TAP's role in supporting biotech companies developing much-needed innovative therapies for patients with rare blood cancers. My name is Jun Xu, Executive Director, the Therapy Acceleration Program Lead, a proud 12-year veteran of TAP. TAP was established in 2007 with the primary goal of supporting the ALS mission. For TAP, unmet medical need, it what drives us to invest to make a difference. No indication is too small. While you're waiting for the event to begin, you probably have noticed that 3 therapies in our portfolio have been approved by regulatory agencies, and the 4th one was included in NCCN guideline for T-cell lymphoma. The bullseye figure on the right captures all the biotech partners of TAP, but don't worry about remembering all the names, because this bullseye graph and with a lot of other information about TAP are readily available on our website. I encourage you to visit our website regularly for all the updates. Before we invite our panelists today who are representatives from BioEvent, DreamBio, ImmuneOnc, and Kymera, I would like to take a few minutes to give you a past example of TAP supporting biotech companies for rare blood cancers. One of the approved therapy TAP supported is Alzheimer's SL401. So this slide shows the journey of SL401. The research grant funding from ALS supported the discovery of SL401 and the early clinical investigation led by Dr. Arthur Franco, an academic researcher. A young biotech in Manhattan named Stemline Therapeutics then licensed SL401. Licensing an intellectual property from academic institutions is very typical how a biotech starts. I still remember the day the company came to us. The team was led by CMO, the Chief Medical Officer. They came to us in December 2012. They asked TAP to support a trial for patients with blastic plasmacytoid dendritic cell neoplasm. That's my early days of TAP, so I was not aware of the disease. And also it took me a while to keep all the 5 letters in place because the name is too long, so we just call it BPDCN. BPDCN is a very rare type of blood cancer with only a few thousand patients globally. However, the disease is very aggressive and with very poor outcomes. What's more, there was no standard of care back then. Patients were treated by physician's choice depending on which doctor the patient went to see. So in December 2012, when the company met us, they told us that a recent preclinical study by a French group has indicated that the BPDCN might be very susceptible to SL401. So we have a cartoon on the right to show that. The cancer cells of BPDCN cells carry CD123 on their cell surface, and then SL401 will bind to the CD123, and then cells will be killed by the toxin SL401 carries. So in 2013, TAP got approval to provide more than $3 million to support the pivotal trial. In the meantime, ALS also has started a very comprehensive education program for both healthcare professionals and patients about the disease. The interim result from the trial was very, very encouraging. So FDA granted the therapy breakthrough therapy designation in 2016 before the trial completed. So after the trial completed, the company filed for approval. So in December 2018, the FDA approved Elzonris for BPDCN for both adults and children. So Elzonris SL401 became the standard of care. As many other cancer cells also highly express CD123. So this therapy is also being investigated for a range of other indications. Let's roll the video clip of Dr. Arthur Franco, who with LS grant support discovered the molecule, and also representative from Stemline Therapeutics, Dr. McDonald.
The Leukemia and Lymphoma Society is critical in that it provides the seed funds to come up with new treatments for treating leukemia and lymphoma and associated diseases. And without that early support, it would be very hard, even with the National Institutes of Health, to make the advances. And they were also willing, and they have a program, to continue to support, once a drug is made, to do the translational work to show that it really works. And I think that for patients that have to undergo chemotherapy, lose their hair, have nausea and vomiting, the development of new drugs with fewer side effects that's aided by the Leukemia and Lymphoma Society is my passion in life and their passion as well.
Leukemia and Lymphoma Society's TAP program, or Therapy Acceleration Program, really helped us build awareness of our clinical trial. The disease that we treat, BPDCN, has a small patient population. It's about 500 to 1,000 patients, and they really helped us build awareness of the disease, build awareness of our drug, and help enroll in the clinical trial to make sure that this patient or this drug got out to benefit the most patients as possible. The Leukemia and Lymphoma Society is a very important part of our work. It helps us reach patients, it helps build awareness, and does a lot of good work funding diseases for patients who are in need. We would tell anyone who's looking to enter into the Therapy Acceleration Program that it's an excellent opportunity to highlight your research. It's a rigorous process. It builds a lot of credibility and visibility with both investors, patients, and researchers.
I hope you have enjoyed the video. I have watched the video, of course, multiple times, but every time I watch it after that, I still feel like, wow, what a difference LS has made from a discovery to approval of a new therapy, and that is a standard of care. Without further ado, let's hear how TAP is working today with other biotech companies to tackle other rare blood cancers. Just a reminder, you can use the Q&A feature to post your questions to the panelists, and we'll try to address them at the end of the program. And also, this event is being recorded, and the recording will be available afterwards. First, welcome my TAP colleague, Laura and Blaine.
Good afternoon, everyone. It's a real pleasure that you're joining us today. So my name is Laura Grunbaum, and I'm leading our Therapy Acceleration Program here at LLS. And together with my colleague Blaine, we'll be having hopefully what will be an interesting and really informative conversation on what's happening for rare diseases. So before we go there, Blaine, do you want to briefly introduce yourself?
Yeah, thanks, Laura. Happy to be here today. Hi, everyone. I'm Blaine Robinson. I'm also an executive director with the Therapy Acceleration Program. I've been here with LLS over 13 years now, working closely with June and Laura and the rest of the TAP team to help make decisions for investments, and also working closely with our partners, providing connections and advice and having them succeed in their trials. So let's get today's panelists right up. First, I'd like to invite our first panelist, Jared Golub from Kymera, to please turn on your camera and join us. Hi, Jared. How are you doing today?
Great. I'm doing well. Thanks, Blaine.
Great. Thanks so much for participating in our panel today. So before we get to some questions, please introduce yourself and tell us a little bit about Kymera.
Sure, happy to. So I'm Jared Golub. I'm the Chief Medical Officer at Kymera. I've been here for about 5.5 years. Kymera was established back in 2016. The company went public in 2020. We're located in Watertown, Massachusetts. We have about 200 people working here. We are a platform-based company. We focus on developing targeted protein degraders for targets of interest across multiple different disease areas. These are targets that are either undrugged or inadequately drugged. And essentially, we have moved multiple programs into the clinic over the past 2 to 3 years, 4 programs in total, including both oncology and immunology programs. We currently have several oncology programs in phase 1 in the clinic in our pipeline, and we have an immunology program that's partnered with Sanofi that is currently in phase 2 trials.
Great, thanks, Jared. So for just people on the webinar, so Kymera has been a TAP partner for almost 4 years now, since early 2020. And obviously, when we made an investment in Kymera, we were very interested in your platform technology. As you mentioned, you're developing first-in-class protein degraders. Several of them obviously were in blood cancers. And as you mentioned, 2 are in the clinic today, specifically in these blood cancers. So I'd like to first start talking about your STAT3 protein degrader program. Congratulations, it's received Fast Track designation for both, CTCL and PTCL. So, before we get into some of the data, interestingly, STAT3 has been around for a while. A lot of people have gone out different approaches and companies. So tell us a little bit why Kymera went after STAT3 as a target and how your degrader differentiates itself from others that have been developed.
Yeah, thanks, Blaine. Yeah, as you said, STAT3, has garnered a lot of interest both in oncology and immunology over the years as a transcription factor. In oncology, it's known to drive proliferation and resistance to cell death in certain tumor types, especially certain liquid tumor types like lymphomas. It also confers resistance to various targeted therapies and chemotherapy and radiation therapy, and also has an important role in setting up an immunosuppressive tumor microenvironment that can lead to resistance to drugs like immune checkpoint inhibitors. So Kymera, we were very interested in this target for those reasons, and also the fact that because it is a transcription factor, it's very difficult to drug with traditional small molecules. You want to be able to get rid of both the phosphorylation-dependent and independent functions of STAT3, and you also want to be able to be highly selective because hitting other STATs can have real safety issues, and you also want a drug that's potent. And we found that we could develop a degrader that really accomplishes all 3 of those things. It's very potent, it's highly selective for STAT3, and as a degrader, it gets rid of the protein as opposed to just partially blocking its function. And that was the reason why we chose to develop this approach for going after this sort of undrugged or inadequately drugged target and moving the program into the clinic in high unmet need liquid and solid tumors.
Yeah, excellent. Yeah, it totally makes sense. And very exciting from the recent, American Society of Hematology meeting just in December, you presented your STAT3 clinical program, the initial results. Can you tell us about those results and where this program's headed?
Sure. Yeah, I mean, those were very exciting results for us. We're currently in phase 1A dose escalation. This study is broadly enrolling both solid tumor patients as well as liquid tumors, including lymphomas and leukemias. At the time that we presented those results at ASH last year, we had enrolled about 29 patients across the first 5 dose levels, and we're very encouraged to see that we were getting very robust knockdown, more than 85–90% knockdown of this target in blood and also in tumor that we were able to measure in tumor biopsies, and that was associated with a very favorable safety profile. Importantly, we were also seeing tumor responses, including major responses in cutaneous T-cell lymphoma. So, for example, we had treated at that time 5 patients, and we're seeing 2 partial responses and 1 prolonged stable disease. And we also saw a very good partial response in Hodgkin's lymphoma, in addition to seeing prolonged stable disease in certain head and neck tumors. So for us, it was very exciting to see this sort of activity, some of which was playing out the way we had predicted from our preclinical models in T-cell lymphomas, including cutaneous T-cell lymphoma, and some that were of great interest to us because we hadn't explored this in preclinical models, especially the result that we saw in Hodgkin's lymphoma. In addition, we also saw that the drug was clearly having an important immunomodulatory effect. We could see the induction of this so-called interferon gamma response, both in the blood and in tumor. And in preclinical studies, that sort of induction of that type of interferon gamma response is associated with heightened sensitivity to checkpoint inhibitors like anti-PD-1 drugs. And we've actually shown that in a preclinical syngeneic solid tumor model. So for us, that represents really another very important opportunity for this drug to be able to maybe in solid tumors combine this drug with either anti-PD-1 drugs or other targeted agents. And given the activity that we're seeing in these T-cell lymphomas and Hodgkin's lymphoma, an opportunity for this drug as a monotherapy to be developed in these high unmet need areas.
So Jared, of course, the focus today is on these rare indications and how to approach this. And I'd like if you can tell us a little bit more about how Kymera is thinking in terms of developing a strategy, which of these rare indications to include, and there's a whole slew of them, as we know, when we look at T-cell malignancies, how to really approach those populations, how to build up a strategy that allows you to enroll patients in a timely fashion and to accelerate a program and hopefully ultimately move it to an approval in the future.
Yeah, I mean, I think that's a really important question, Laura. I mean, initially, we want to be data-driven in terms of the type of indications that we go after with any of our programs. So our preclinical work, in T-cell malignancies in particular, pointed us toward, PTCL and CTCL. The fact that LGL leukemia, that you see activating mutations in STAT3 in over half of those patients, was also pointing us in that direction. But to your point, when you're going after these relatively rare lymphomas and leukemias, one has to have a strategy, for being able to find those patients. I mean, I think it's clear that across those different diseases, there remains high unmet medical need, either in the relapse refractory setting or in some cases even in the frontline setting. And there haven't been a whole lot of active drugs that have been developed in those spaces. So for us, it was important to align ourselves with investigators who had these patients, because while the study is broadly open to patients with solid tumors and lymphomas and leukemias, we made a point of going out and selecting sites that we knew also had expertise in cutaneous T-cell lymphoma, peripheral T-cell lymphoma, and LGL leukemia. And so fortunately, we continue to be able to enroll these patients onto the study and gain experience. Because ultimately, I think once we complete phase 1A dose escalation, which we're planning to do this year, it'll be a combination of looking at all of the totality of the clinical data in terms of where we think we're seeing activity, and then our preclinical data, where we look at our drug as both a monotherapy and as a combination therapy, and use all of that information together to then decide on the next stage of development. We want development in phase 1B and phase 2 and beyond to really be driven by, not just the pharmacodynamic activity and the safety, but importantly, what sort of activity are we seeing in the clinic and in our preclinical models to really, de-risk whatever we decide to do in phase 1B and phase 2, especially in these select rarer populations of tumors.
Yeah, I think we're going to hear more about these challenges and how different companies are approaching that. So maybe just a very quick note, because of course you have a larger portfolio and we see there's probably something else in the blood cancer space that we all should be paying attention to at Kymera. Do you want to just provide a little teaser to our audience?
Yeah, absolutely. We have a second program which is also in the clinic in phase 1A dose escalation, which is targeting MDM2. And this is a target that people have been interested in for years. And there have been a number of small molecule inhibitors that have been put into the clinic, with varying, efficacy and results in both liquid tumors like AML and in certain solid tumor types. And one of the real limitations of using small molecule inhibitors has been a feedback loop that leads to upregulation of MDM2 and blunts the ability to upregulate and activate P53, the tumor suppressor, which is what you need to do to then drive cells to apoptosis. By using a degrader, we can overcome that feedback loop, and we have some very exciting preclinical data showing substantial activity and potency greater than small molecule inhibitors across various solid tumor types and AML. And we're now in the clinic with this drug. And last November, we actually shared some of our early data in our early dose levels, where we're already seeing, a major response in Merkel cell carcinoma, which is a neuroendocrine tumor that we expected from our preclinical models to be sensitive to a degrader. And so we saw an impressive response even at our first dose level. We have been seeing clear evidence for P53 pathway activation with our degrader. And importantly, we're not seeing the typical myelotoxicity that one sees with small molecule inhibitors with our drug, which is given intermittently as a once every 3-week infusion. And so we're excited that we think we have an approach to targeting MDM2 that can avoid the typical dose-limiting toxicities you see with small molecule inhibitors, and may open up, our ability to really develop this drug effectively in liquid and solid tumors, and also to more effectively select which types of patients, especially solid tumor patients, who are going to be responsive to this treatment. But on the AML side, we've also shown some very interesting data that our drug can be active even in patients who have gotten prior venetoclax. And so we are actually looking at our drug both as a monotherapy in these patients, but also potentially as a combination therapy. So I think a lot more to come, I think, this year for this program as we share more data, but it's one that we're also very excited about.
Yeah, thanks so much, Jared. Really appreciate it. We need to move on to our next panelist, so please turn off your camera, Jared. We'll talk to you later in the Q&A. I'd like to invite next panelist, Matthias Will from Dren Bio, to please join us. Hi, Matthias. How are you doing?
Excellent. Thank you. Thank you for inviting me, Blaine.
From the West Coast today. So glad you're up early and joining us. So before we get to some questions, could you please introduce yourself, Matthias, and also your company, Dren Bio?
Sure. Yeah, Matthias Will. I'm the Chief Medical Officer at Dren Bio. I joined the company 2.5 years ago. And Dren Bio is a privately held biotech company. We are based here in the Bay Area. We are smaller than Jared's company, 65 people strong. And we were founded in 2019 by Nina Tomasovic. I mentioned the company is privately held. TAP joined the company as a strategic investor with our Series B financing. That was in 2022. And since end of 2021, we also have a partnership for 2 preclinical programs with Pfizer.
Great, thanks so much. Yeah, as you mentioned, right, sort of on the other spectrum, you're a relatively new partner with TAP, a little over a year now. But like Chimera and others today, we were very interested in the programs you were developing. You have a very interesting monoclonal antibody that's going after a unique target in rare blood cancers, which we'll talk about briefly. In addition to you've developed a bispecific antibody, a novel platform, which in both aspects is very encouraging, and we wanted to learn more about it. Hence the partnership. So let's first talk about your DRO1 program. If you tell the audience, what the program's about, what target you're going after, and the current status of the clinical trial, that would be appreciated. Thank you.
Sure. Yeah, so as you mentioned, Blaine, we have currently 2 programs in the clinic. Both of them are targeting hematomalignancies, and DRENO1 is the most advanced one of the 2. We are at the end of our dose escalation. We have enrolled about 30 patients right now. And the target is truly unique. We are the first targeting CD94. CD94 is a scaffolding protein. It doesn't have an intracellular signaling domain. And it's a dimerization partner for NKG2A and NKG2C predominantly. As such, it is really restrictively expressed on CD8 T lymphocytes, and they are only on a subset of CD8 T lymphocytes, which are the terminal differentiated effector T lymphocytes, and on NK cells. So taking this together, it is really a specific selected target on cytotoxic cells. And as such, our initial indication is also restricted in the hematologic space to these CD8 and NK cell-driven lymphomas and leukemias. When you think about T-cell lymphomas, as Jared mentioned before, they really fall into 2 larger buckets of cells of origin. The CD4-driven ones, which are the probably more commonly known ones, like your PTCL NOS, your angioimmunoblastic T-cell lymphoma, ALCL, and so forth, including the cutaneous T-cell lymphomas like Sézary syndrome and mucosis fungoides. The smaller subset, about 20% of the T-cell lymphomas, are really CD8 and NK cell driven. And amongst those, you find some of the most aggressive T-cell lymphomas with really abysmal prognosis. Common to them, and there are more than 12 different histological subtypes described by the WHO, is that they are driven by cytotoxic cells, either CD8 or NK cells. And also, only 2 of them have actually an NCCN guideline designation about a frontline therapy. None of them have any standard of care defined for the relapse or refractory situation.
Matthias, thank you for illustrating how challenging it can be in this space. And I wanted to ask a couple questions, again, related to the specific hurdles that you have to overcome. One of them being, how do you really pursue these very, very rare patients? And then the other one is related to, an associated problem, which is often that it's hard to get even international alignment on treatment criteria, on diagnostics. And I know you've made some efforts in this space to really bring together KOLs internationally to really get some alignment on these parameters. And of course, we all know ultimately they're going to be very important to define what's really required for an approval and to have a productive dialogue with FDA or with regulators worldwide. So anything you can share with our audience on these challenges and how you're approaching these would be valuable.
Yeah, thank you, Laura. As Jared mentioned, and I want to dovetail there, it is fundamentally essential to work really with the right investigators and key opinion leaders in this space. The good news is, given the rarity, most hematologists who are not an expert in this field are happy to refer these patients to expert centers. So it comes down to an overseeable number of investigators to work with in each of the countries. Our study currently is open and enrolling in Australia and the United States, but we will be expanding very soon also into the rest of the world, into Europe and Asia Pacific. But as you mentioned, treatment options differ. Not all agents are approved in all the regions. And also the response criteria evaluation or even the diagnostic criteria are not necessarily well established. I give you one example or maybe two. One is hepatosplenic T-cell lymphoma. Typically, you would evaluate an aggressive lymphoma that is PET-AVID with PET-CT and with Lugano criteria. But this lymphoma has its primary location in the spleen and the liver. Now, for a PET-CT, you will need your reference point as the uptake in the liver. If your predominant manifestation of a lymphoma is in the liver, obviously this will not work. So there is a triangulation to be made around an alignment with investigators. What would be the right approach to actually evaluate qualitatively and quantitatively the response in this malignancy? And then eventually also get your different regulatory bodies on board with how you think about this. So that's certainly a challenge with rare diseases and hematomalignancies. And another example would be, for example, LGLL, which Jared already mentioned, where also very few, thinking of, there are 3 prospective trials I'm aware of, 1 ongoing, 2 completed. Are the inclusion criteria somewhat comparable? Are the response criteria somewhat comparable? And how can we eventually, even for these rare diseases, strive to a standard?
Yeah, absolutely. And of course, this is an area that is really near and dear to our hearts here at LLS, and where we really want to help, through partnership, through help with education and communications with broad audiences, all the stakeholders involved, so that we can help to get this alignment and get the messages out that are important for the physician community or patients overall to understand.
Yeah, so in the year we are collaborating together, I mean, you have been obviously instrumental in helping and guiding us there, and particularly as we progress and thinking about how would we get this to a broader patient community, obviously your role will just increase for us.
So maybe I'll also just say a quick sneak peek at the platform that Blaine mentioned, which also is exciting because it has potential for a range of indications, including blood cancer. So just the, very, very quick download. How is this different? How is this exciting?
Yeah, so while our first program targeting CD94 is a non-fucosylated monoclonal antibody, our platform is a bispecific platform. Obviously, there have been a lot of excitement recently in the biologic space, both with the next generation ADCs and solid tumors, but then also the T-cell engagers, bispecifics for lymphomas. Now, our approach is fundamentally different. We are using the bispecific to really cross-link, in our first case, lymphoma cells with myeloid cells, and inducing a strong phagocytic signal, an activating signal, which leads to engulfment of lymphoma cells and eventually then phagocytosis and digestion. And in a second step, then these myeloid cells, as professional antigen presenters, also present the subset of the digested cells antigens to their surface, release cytokines, and attract and activate T cells. So there is a second MOA aspect, which is really inducing adaptive immune response after the phagocytic event. And this study right now just started as well in the clinic, and we are very curious and excited for the first results as we are enrolling patients throughout this year.
And so are we, of course. So thank you, Matthias. And I'd like to now invite our next panelist to please come on, Andres McAllister from BioInvent. Andres, welcome. So why don't you go ahead and tell us a little bit more about your role at BioInvent and what BioInvent is doing?
Super. Thank you very much, Laura. Thank you, Blaine, for the invitation. My name is Andres McAllister. I'm Chief Medical Officer at BioInvent. BioInvent is an immuno-oncology-focused company, really trying to understand and help patients through the manipulation of the immune system. The company was started several years ago in Lund, in the southern part of Sweden, where we have our headquarters, where we have all the great majority of the people who work for us. And we are a mid-sized biotech in the order of 120 full-time employees at this stage. So happy to be here. I'm happy to tell you about our programs.
Thank you. And of course, we are proud to say that you are actually our 9th European partner company. So here at LLS, we've recognized a long time ago already that there's a lot of innovation, a lot of biotech energy and drive is coming from outside of the US. And one of our goals over the past couple of years has really been to help bring this forward more quickly, bring it to patients worldwide, including here in the US where we are based. And some of you may have seen our panel event last year, which was specifically focused on how we help to accelerate through partnership with European partners, these therapies to come to the clinic and hopefully ultimately to come to approval worldwide. So earlier this year, your company announced some strategic priorities for 2024. And we were, of course, very happy to see that 2 blood cancer programs are top of your list. And one of them, BI-1808, is your most mature program, I'll say, and you had very nice results presented at SITC in November of last year. So we would love for you to tell us a little bit more about this program and tell us of the results that you've seen and what you plan.
Sure. So BI-1808 is an antibody. I'll explain in a minute how we find antibodies, etc. But this is the final outcome is to find a good antibody toward a good target, and that's what we think we have. So it's a monoclonal antibody, an IgG1 antibody, fully competent, that recognizes TNF receptor 2, and TNF receptor 2 is, of course, a receptor for TNF, but it has the interest that it's very highly expressed in the tumor microenvironment and in particular by Tregs. So we've done a great deal of characterization, and we see, for instance, that in animals, but now in patients as well, we see that when you give this antibody, you basically decrease the numbers of Tregs significantly, and that actually we're starting to see that when you do that, you actually can have clinical responses. So I think that's what's most interesting in that clinical. So the antibody right now is entering phase 2A. We started dosing and went up to a high dose where we measure receptor occupancy. We measure a number of different biomarkers, including, for instance, soluble TNF receptor, and a number of different things, populations of cells that change, etc. And what we see is that we're starting where we are right now. We have a very nice occupancy of the receptors. We see the same changes that we saw in preclinical studies. We're seeing them in human beings, and so we're super excited. And of course, the question is, how is it doing anything? And I think we're starting to see those first signs of efficacy. We had this very interesting patient with a solid tumor with GIST that had a significant reduction in sort of partial response with very solid reduction in all the lesions, and some of the metastatic lesions disappeared, etc. So very exciting times in that patient. For instance, in particular, we see the highest drop in Tregs. So that's very exciting because it really shows that what we're trying to do, it's actually happening in patients. So in one of the things that is interesting about TNF receptor 2 is that it's highly expressed by several T-cell lymphomas. There is a body of literature out there showing that, and so we think that right now we started out thinking about CTCL, but we're actually making that larger and just throwing a wider net and trying to see whether that will work in T-cell lymphomas. And so that's where we stand. We've started to treat the first few patients, so it's very early days, but looking very interesting also on that front. So that's where we are with the TNF receptor 2 program, mainly BI-1808.
Right, thanks, Andreas. Yeah, so we're looking forward to the data as it progresses with both the CTCL and as you expand it in T-cell lymphoma patients. Now, we've been partners for just about a year now, and we here at TAP, feel like we've heard we have a very rigorous due diligence process, and as part of the process, we use world-leading experts and clinicians as some of our advisors to help evaluate the process. And during our collaboration, can you comment on some of the interactions you've had with some of the KOLs that we've introduced you to, and how that's produce some clinical activity and to increase your enrollment in the trials?
Sure. So Blaine, I think this has been when as you mentioned, the due diligence process and being confronted with very prestigious, knowledgeable folks out there. It was actually an opportunity for us because the company really prides itself in producing high standard science published in the best journals, etc. So it was actually an opportunity to interact with them and show them the quality, and I think they did recognize it, and the great outcome of this, and thank you for that, thank you LLS for that, is that we've now engaged several major investigators in these areas, and they've actually now they're actually recruiting patients in our studies, and without that, I doubt that a small company out of Lund in Sweden would be able to execute those clinical studies. So as 1808 is open in the US, we have several sites already, and they're recruiting patients that absolutely will allow us to look whether this is a helpful therapeutic candidate for patients with T-cell lymphomas and CTCL.
Yeah, great. Yeah, looking forward to the data as it comes out. So obviously a European company, so you've got trials active in Europe and also in the US. We understand you're planning clinical trials in Latin America and South America. Maybe you could tell us a little bit about that process, how you see this as a potential opportunity to enroll in the trials, and if there's any sort of challenges in trying to get these sites up and running, and what types of patients can you expect to see in those areas of the world?
Yeah, so it's a very interesting question. So we've been looking, of course, recruiting patients in some of these indications is very hard, as we've already heard it from the other panelists. And definitely our feeling is that Latin America is an unexploited, or at least not as exploited place where you can find those patients. And actually, we've been looking seriously at places like Brazil, Colombia, Chile, Argentina, etc. And what we find there is that actually a lot of the clinicians are US trained, European trained, so they have a perfect understanding of what's happening, etc. So my first impression is that it is very positive. It is definitely something that should be explored, especially if you're looking for those rare patients. They traditionally larger pharma companies go there for the purpose of running their phase 3 studies, sometimes phase 2 studies. But these early years of development, which is, I think, the most challenging part, they usually don't get to do that. So they're very excited and very they're happy to see that that someone is thinking about doing that over there. In terms of challenges, so I think from the clinician standpoint, from that end, I think there is really not that difficult. I think the difficulty comes sometimes from the time it takes to approval, etc. So running having a robust support regulatory wise, I think that's helpful. So, but in my interaction with people who've done the process, I think everyone at the end of the day is very happy because you tap into getting access to patients that you otherwise wouldn't have in places like Europe or even the US.
Absolutely, Andres. Thank you very much. And that's a unique perspective, I think, that you can give, and hopefully something that's encouraging to other companies as well to consider these countries and not to dismiss them upfront, that innovation is possible in many places and we really have to push for it. So thanks again, Andres. And I'd like to come to our last panelist today, Charlene Liao from Immunonc. Charlene, if you would please turn on your video and sound. Welcome, Charlene. Now, you, of course, have a long relationship with LLS. So I'll let you introduce yourself and tell our audience a little bit about Immunonc and what you're up to.
Yeah, I'm Charlene Liao. I'm the founder, CEO, and board chair of Immunonc Therapeutics. Immunonc Therapeutics is discovering and developing first-in-class therapeutics for patients in blood cancer, solid tumor, and autoimmune diseases. We are established in 2016, and we are headquartered in Palo Alto, California. As you can see, the Golden Gate Bridge behind me. We build on fundamental sciences with a target focus on inhibitory receptors expressed on myeloid cells. Initially, the immunoglobulin-like receptor B families or LILRB families. From our foundational science, we have advanced several programs at a different stage of development. Our IO-108, for example, is targeting LILRB2 for solid tumors, and it's currently in phase 1B expansion, and we hope soon will enter phase 2. And our IO-202 is targeting LILRB4, and it is in phase 1B development for monocytic acute myeloid leukemia, AML, and chronic myelomonocytic leukemia, CMML.
And of course, we've had, as I said, quite a history with your target RB4 as well. We actually supported through our grant program early research by Dr. C.C. Zhang of the University of Texas Southwestern Medical Center, who developed an initial antibody against RB4 and demonstrated also the potential for utility in the medical space. Your company acquired the antibody, and you've been a partner with us since 2021. We continue, of course, to be excited to be working with you and supporting you. So you mentioned 202, so that's quite a unique program. So tell us a little bit more about the target and also how you choose. You mentioned CMML, that is a very rare indication, which of course is front and center today. Tell us a little bit more how you came to choose this indication and what you're doing there.
Yeah, thanks to LLS's support to academic researchers, and we are very proud to have Dr. Alex Zhang as our scientific founder. We together have published the paper in Nature to elucidate the role of LILRB4 in acute myeloid leukemia. Basically, LILRB4 as a target is found specifically on a subset of acute myeloid leukemia called monocytic AML, as well as myelomonocytic AML. Because of the specificity of this target, we are able to develop an antibody that is the IgG1 subtype, which we advanced to clinic. And this antibody, IO-202, is the first-in-class antibody targeting LILRB4. We know acute myeloid leukemia is an orphan disease with roughly 20,000 newly diagnosed cases in the US every year. And within AML, there are a few subtypes. One of them is the monocytic AML, representing roughly 10% of the total AML. Because monocytic AML expresses a high LILRB4, we are able to show in the clinic the ability to specifically removing leukemia blast, leading to complete responses in a patient. And monocytic AML is also in need of better treatment because it has been shown by LLS supported clinical group that it is resistant to the newly approved medicines such as venetoclax and azacitidine. So we really have a very good hope for IO-202 to be able to address a specific AML subset. Now, speaking of CMML, it is more rare. It has the incidence rate of about 1,100 cases per year in the US. And we found also LILRB4 is highly expressed on CMML blasts. And we have very nice data showing the effectiveness of IO-202 in CMML.
And of course, we at LLS are very committed to help develop better therapies for CMML. And we've really received an incredible boost through a very generous donation of $17 million from the Segal Family Foundation. And this funding includes not only a grant program of $13 million, and we're very happy to say that 7 grants are actively on the way, but also support through potentially the TAP program that we are discussing today, education programs, and a range of other activities to really help accelerate enrollment of patients, awareness and enrollment, because this is so critical for these rare indications and for enrollment.
Yeah, and Charlene, I know you mentioned, 202, it's in the clinic now. You presented some of the first initial results last year at the European Hematology meeting. Can you please share our audience some of those initial findings and sort of the current status of the trial and what types of patients you're looking for, any particular challenges you're having? I think our audience would appreciate that.
Yeah, thank you, Blaine. We're very pleased to present the IO-202 dose escalation data at the European Hematology Association Congress in 2023. We found that IO-202 has demonstrated a strong safety and promising clinical activity, either as a monotherapy or in combination with hypomethylating agent azacitidine. And both in relapse refractory AML as well as relapse refractory CMML. This suggests that LILRB4 may be an important therapeutic target for hard-to-treat blood cancers. So our clinical data support the mechanism of action of IO-202. As I mentioned, IO-202 is a humanized IgG1 antibody, the type of antibody with ability to induce antibody-dependent cellular cytotoxicity, ADCC, and antibody-dependent cellular phagocytosis, ADCP. And in fact, ADCC and ADCP are target copy number dependent. We found the higher copy number of LILRB4 and the higher percentage of leukemia blasts expressing LILRB4, then the better the killing of these malignant blasts by IO-202. And these are precisely the patients with significant unmet medical needs. So here we have a precision medicine approach and a targeted therapy with IO-202. Currently, we have a CMML expansion cohort open, and we are actively looking for newly diagnosed hypomethylating agent naive CMML patients to enroll in our trial. We're very glad to see the patients enrolled in the expansion cohort so far have all shown very nice responses with immediate marrow complete remission in first cycle of treatment, and some with subsequent complete remission achieved. So we are really pushing the boundaries and transforming lives for patients with rare blood cancer.
Thank you, Charlene. I think we are coming closer to the end of our event today. So I would like to move on to our Q&A portion, and I'll ask all the other panelists to please turn their cameras and microphones back on. And we've hopefully now seen that there's a range of approaches how you can really impact clinical development for rare indications. And while we've been speaking, hopefully you in the audience have been asking questions, and there's still an opportunity to continue to do that. So please use the Q&A box. But I'll call on my colleague, June. June, do you have anything there from our audience for our panelists? Any questions?
Thank you, Laura, and thanks to all the panelists. It's a really wonderful session. So there's a few questions about how TAP works, very specific questions. So I encourage people to really go to our website. There's a lot of information there. You can simply Google LLS TAP, then you'll find our website. So I'm not going to really have us or the panelists address those specific questions, but we do have a general question, for the panelists, because we talk about rare diseases. We all know the hurdle you touch upon, here and there. So if you have a wish list, what are your top concerns in terms like getting it approved? So if you want to tell something to the regulatory authorities, right? So what will be sort of your wish list?
Let's start with what's rarest here, which may be the cytotoxic lymphomas, T and NK cell lymphomas. Matthias, any thoughts?
Yeah, I probably would say a comprehensive approach and dialogue with regulators about what defines standard of care and what doesn't define standard of care. That would be something, because it obviously has a significant impact on the sizing of a study and the expectations towards the study. And as we said, these patients are rare, and there is the line we all need to walk with. How much certainty do we get about the true insights about standard of care versus in some cases we need to basically draw a panel and basically say what are the leaders in the United States, for example, using, and then rely on that. So I think that would be helpful.
Mm-hmm. Mm-hmm. I'm sure we have more perspectives. Maybe Jared, to stay on the T cell side.
Sure. Yeah, I think I think our hope is that regulators, both in the U.S. and Europe, would continue to have an open mind regarding the sort of where the bar is set regarding initial approvals for these rare cancers, especially still leaving the door open for sort of single arm sort of accelerated approvals. Obviously, for rare cancers, whether it's CMML or LGL, or even different subsets of CTCL and PTCL, right? There aren't large numbers there, and so the expectation that there are going to be randomized controlled studies or large studies to get initial approval, if you have a drug that is highly active and well tolerated, our hope is that there are paths still available to accelerating development and getting drug to these patients, provided you do have convincing data with regard to efficacy, and that safety is clearly acceptable.
Mm-hmm. Charlene, do you want to add from the CMML perspective? I know you guys are actively thinking about what's required to make it all the way. too.
Right. Yeah. CMML is a rare leukemia, as I mentioned, and in terms of standard of care, the currently approved agents are hypomethylating agents, but it was approved in the context with other diseases, with more patients in a trial having MDS, and only a dozen also patients with CMML. So it is hard to conclude what is the rate, benchmark rate for the standard of care. So we need to have a constructive dialogue with the regulatory bodies on what are the rate, the bar for success. But another very important hurdle for us is generating the most impactful data through clinical trial. And we really appreciate Leukemia and Lymphoma Society's support in patient education as well as physician education, bring big awareness to the CMML as a distinctive disease and distinctive from other myeloid dysplasia or malignancies. So patient education, patient outreach is crucial for us to successfully enroll in a clinical trial.
We definitely agree. That's been our experience, as I think our audience has seen with the story of BPDCN and Elzonris. Andres, anything from the European perspective that we should consider?
Yeah, well, I'd like to say that obviously next to what the other panelists have said, I totally agree with that. But also, if I had a conversation with the regulators, I'd like to tell them that think twice about lifting the bar, because when you lift the bar, you hit very hard innovation. Innovation is the most difficult thing to bring forward. And if you lift the bar, so you start asking the same things of small biotechs to big pharma, you're basically killing innovation, or at least you will give it a big hit. So I think when they lift the bar and they request to do studies that are much bigger than originally thought and understood. They want to protect the patients, etc. But you should really think hard about that, because you can hit very hard innovation, and that's the first thing that suffers.
Yeah, thank you for that. I think we all agree. It's not one size fits all. We really need to have that detailed dialogue, and I think there's a very good understanding of the regulator side as well, and they really welcome and encourage the dialogue and are providing increasing opportunities in that area. So let me go back to June. Maybe we can do a lightning last question, or have we addressed most of what's been coming from our audience? And TAP related, we can, please contact us independently. There's contact information on our website.
Right. I think we have more specific questions. For the interest time, I think we can answer them later on, address them.
Okay, that sounds good. So hopefully, thank you, first of all of you panelists for spending this time with us and sharing what you're doing, your insights and your wish lists. So I will let you go off to your daily work, which we really value very much. And thank you to all of you who have been joining us and who have stayed on. We hope this event has given you some food for thought and also been a source of some optimism that things are moving for rare cancers. And of course, we at LLS will remain committed to do this and to really try and help and achieve better outcomes and cures for all patients with blood cancer. You can also meet us in person. We are at all major conferences, scientific conferences, biotech conferences, and we hope to be back with another interesting panel discussion next year in January, post JP Morgan. We'll be in touch with you. So please check us out on the website, and thank you again. Enjoy the rest of the day.
Thank you.